Seems like it's working. So let's continue. So please ask your questions in any language you can. I will try to understand and reply. And also, don't be afraid of asking questions on most of slides, just to make your feel what this proteomics is and to see what you already know and what we can skip ahead. There is unfortunately no single book on proteomics that you can use, but I think at least those 3 books are usable, and also everything that you will need on a final test will be in our lectures. So not a problem if you will have no such books. But still, I do recommend this nice book from TA, Genomes. It's a 4th edition right now. And also, this book is available through the Sci-Hub. It's possible to get it, Introduction to Proteomics. Unfortunately, this is a copyrighted book. I can't put it on a Google Drive. It will say that it's a copyrighted book, so unfortunately not. And this book is also available through the same Sci-Hub. So, our plan for 2 weeks, we'll have 2 lectures every day, with the exception of Mondays. And next week on Friday, we'll have some practice work. So, if you want, you can bring your laptops, and we'll just try to do step by step some analysis of data, so you can actually try to do it by yourself. Because for most part, if you will be working not in Russia but in some other country, all you will need to do is to get your sample, to treat it with some chemicals and some other agents we'll be talking about them, get it to some core facility, and after this you will get just some files of data. And we'll be, of course, discussing what this core facility will be doing to your sample, but mostly your work with core facilities, you get your sample and get data back. And then you're on your own with this data.
Have you worked on these facilities?
I worked at St. Petersburg State University core facility, research facility for cell molecular biology, since it's organizing for 4 years, but have to leave it because there were some problems with administration, but still. So I can get you some data from the first hands how it is done, at least some 4 years ago. So at September 13th, is it Friday? No, it's not Friday. We'll have student reports. Because at 19th we will have some topic assignment, and we will have to get some short, 10 minutes at max, report on a topic that you will choose. So if you have some different topics aside from which I will be giving to you, you can also discuss, and we can add some topics to this list. And we'll have our test online using Google Forms. I will just send you a link, and you'll have more than a day to complete this test. And there is no problem if you will need to consult with literature, with textbooks, with everything, because it would be not the checking of your memory, but of how you can use this data that you already get from the courses. And last Friday there will be final evaluation. If you will have any questions, any discussion about this course, you are welcome. So let's start from 4 questions, just to define our proteomics. Do you think that the genome, even if it would be fully sequenced, by the way, there is no fully sequenced genome as there is up to date, even a human genome, only 1 chromosome is sequenced from telomere to telomere, and unfortunately...
This year there was an update.
There was an update, but it still says that there are some parts which are highly variable, or actually there are some deletion that are in most of populations that are not presented in this genome. So, well, even if you have the whole genome, even pangenome of the whole human population, would it be possible to fully explain phenotype of just a given person?
No,
No.
No. Why?
because not whole genome, I mean, for example, genes are expressed. For
Different.
example, it is the one.
Yes,
the
but you have this blueprint. What is missing? Genome is a blueprint,
Mhm.
and usually if you have blueprint of something which is fully detailed, what material should be used, what should be done, in what order, we can create any machine, right? Why it's not so with genome? What are we missing to get from genome to phenotype?
Proteins.
Not only proteins
mRNA.
per se, and not only mRNA. Does an environment have any impact of how this program would be realized from this genome to this phenotype?
Epigenome.
Not only epigenomic program, right? Because there is a lot of illnesses, of human illnesses, which are illnesses of development. When at some point there is some misdevelopment happened, and then this program should we have everything and the genome is fine and everything is fine, but we still have this illness, right? So there's a lot of interconnection. So just having a genome, even at its best form, is not enough. If you have all the transcriptome, will it be enough?
No.
Even for every cell in an organism, in any given time.
It's not enough. And even if we have all the proteins, it is
It
still.
would be my next question, but let's start from transcriptome. What is missing?
The
So-
interaction between protein, because
First, right? Because we can't get from the transcriptome, from the level of expression of every processed mRNA to the level of proteins, right?
Yeah.
It's impossible just to make this equal sign and just have some let's say equation that such amount of mRNA means such an amount of protein, right? Because there are also a lot of regulation, and some of this regulation depend on environment and on previous state of the system, right? What we actually also missing that even if we have a genome for the organism, a transcriptome for the organism, and even if this organism starts from one cell, like we have the zygote and we have to just have to see how it will be developing, it still have some proteins and metabolites from the mother organism. And it have its own ways of modifying this program. So even so, let's get a little bit closer to phenotype. If we have all the proteoforms in organism, all the localization of these proteoforms, everything, will it suffice to describe the phenotype of this organism? Obviously not, because I have this equation, but what is missing at this level?
So we cannot see the whole picture.
But why not? We have localization of these proteins. We have its quantity, quality, all the proteoforms, whatever. What is missing?
Maybe interaction between.
Yes, interaction, but not only between proteins.
Between cells, between tissue.
Between cells we can derive it actually, but this person or this object is living in some environment as well. And we can't say what this object will have before we take this picture and after we take this picture. For instance, let's say we have some person which takes some medicine. Like we can derive this information of what xenobiotic was added to this person from the proteins, right? Only after some time, organism will respond to this xenobiotic and will have something to actually work on, right? And we came to the next idea. We have to have all the metabolome, right? And even so, if we have the all metabolome, we don't know all the interaction because there is microbiome, there is different ways of regulation of the system. So even at this low level of proteome, we can derive some characteristics of this organism. But yeah.
I have an idea. So, for example, if we imagine that we could get an egg from the women and the sperm from the man and make all the development from scratch, could we still explain the phenotype? I don't know if there is a way to calculate all the all the
Nice question,
environment.
actually. We came to the question of statistics, and we came to the mathematical field of attractors. Have you had a mathematical course,
Yeah.
like linear algebra? So you know about such an idea as mathematical attractors. When you change you change some starting condition of your formula, and it's either you get some results which is more closer and closer to some constant or wider, or even a tiny bit of differences in starting condition leads you to absolutely different picture, absolutely different solution of this equation. So actually, most of our processes work as an attractor which have this turning point, like our homeostasis. Whatever you do to organism, to some extent, of course, it will try to still have this homeostasis. But there is also processes which works as a strange attractor. Just a small change in starting condition will make you end up with just absolutely different results. So actually, even in this case, it's a problem of statistics. Most of the way you will get close enough phenotypes, but there are possibilities. Because you can predict actually if you have only one protein from a complex, and this complex have two different proteins, one protein of this kind and one protein of this kind. It's a statistic whether those proteins will be able to find themselves and actually get to this complex, right? If there's 100 of this protein and 100 of this kind of proteins, statistically it's more or less you have a mathematical equation which get you to some knowledge that, let's say, 90 of those proteins will be having this complex and 10 of them wouldn't be able to find themselves, right? So, okay, we can't explain our phenotype even at this level, but proteome is a little bit closer to the phenotype. Alright. Then another question. There is such a buzzword as post-genomic technology. What do you think? What is a post-genomic technology? Because proteomics is considered a post-genomic technology, and usually when you apply some funds, you write something like you use nano something with post-genome technology with and so on, so on, so on. So what is post-genomic technology?
Our interaction up with the genome, with the things which were produced on the genome level, on genome.
Yes, but you're absolutely right that we use this information. Because in proteomics, we enormously gain from genomics and transcriptomics. Actually, it's our way to search for proteins, to identify them, because we have this annotated databases, right? But what is missing still in this definition of post-genomic technology?
Just after genomics.
Yeah, but still not quite the right answer.
There is no genes. Yeah.
There are genes, believe me. So every year the number in human genome going less and lesser, but still. What do you think? We use this information, right? Genetic information. For what? And what we add to this? What
We use-
were we missing at previous levels?
We use genomic information to annotate the position of the gene. And from this position, we annotate it by transcriptomic and do submit more data for it. And then proteomic is much more accurate.
Not much more accurate. Let's say Proteomics is a screenshot of the process, and you can get the whole picture, right? Because genome is what can happen. Yeah, what could be. Proteome is what is there and what is working. So we add this information, and mostly we interested not in single proteins, but in differences in proteomes, like proteome of diseased person versus normal person, like proteome of stressed plant with plant which grows in normal condition and so on. So we use this information, this omic, and nowadays there's also another buzzword, multi-omics, right? Because we can get the whole information, whole picture for systems biology using just only one layer of this information representation. We have to use all of them and still not get enough results. Okay, then, of course there is such textbook view on biology which is not quite correct, and there is a lot of studies and a lot of articles which starts from the error of central dogma or something, but was there any error in this central dogma? Have you read this short, just 2 pages article which was discussing the central dogma? What was told in this article, actually? There was no mention of RNA, actually. There was no mention of metabolites of phenotype. Nothing. The only that was told there, and what was actually proposed in the central dogma, that the information is stored in DNA and not in protein. And this still stands true, right? Proteins cannot replicate themselves, right? So there is no error in central dogma, but we have to add to this picture those regulatory pathways. That's what different, and that's what is actually at this point of time is true. Of course, probably something will be added to this, but let's see. Genome is with us for the whole life. Still there are mutations, there are some genome editing events, but with some asterisks at the whole life term, it's okay. Then, transcriptome. Transcripts usually have half-life time of some minutes and hours. Let's say that we are talking about eukaryotic cell, so minutes, hours at last. Proteome. Average half-life is hours to days, but there is still an asterisk. And what do you think? Are there some proteins which are presented for longer or for shorter periods of time?
Yeah.
Are there some structural proteins in a cell which we need to just have this form of cell and some transportation inside the cell? What are the proteins which persist here longer in this cell? Like cytoskeleton, right? It is mobile, but just imagine a neuron, right? Also, there are some problematic proteins, like the ones which forms in Alzheimer's disease, and the problem that those proteins cannot be easily degraded, like tau plaques or those Alzheimer's plaques, and they can be there for longer. And also, there are some proteins which have really short half-life. For instance, let's try to memorize some event of cell cycle. Are there any proteins which mark every stage of this cell cycle and control them? Like, are there any proteins which command the start of replication?
Different factors of replication.
Different factors, right? But there are something above them, cyclins, right? And they have a really short half-life, up to minutes. So again, and of course there's phenotype, metabolism, and environment. We will be talking about them, but I really like this idea from the article of 2007, which states that genome is what can happen and how organism can react. Transcriptome is what appear to be happening. Why it's not what's happening, but what is appear to be happening is understandable, right? And proteome is what makes it happen. What tools do we have inside the cell which are available at this time? And metabolism is what already had happened and causes changes, or what is happening right now. So, really nice. You love this nice idea. And of course, we have to add some more levels, like microbiome, like epigenome, like whatever. Still, what was a textbook definition of proteomics? Proteomics actually is not a mature discipline, and it haven't had yet its own methodology and terminology. We use a lot of methods from protein chemistry, from bioinformatics, from genomics, and everything. So, but it starts, I think, in 1994 with Mark Wickens, who had coined this term as the proteome is a total set of protein expressed in a given cell in a given time. He already defines it. It is really easily changeable, so at a given time, and there is a localization at a given cell. So we couldn't have a transcriptome of human person. We can have some pan-transcriptome of different tissues of different humans and so on, but still. And also, what easy to understand here is that we have one genome, but this one genome provides many different transcriptomes. Transcriptome of brain, transcriptome of microglial cell, transcriptome of neuronal cell, transcriptome of, let's say, developing neural cell. It will be different. The one textbook example, which really on many books for proteomics, is insects which have a full transformation. There's a caterpillar from one side and a butterfly from another side. The same genome, totally different proteomes. So, nowadays this proteomics includes different additions, like it's not only identification of total set of proteins, it's not only overall level of protein composition, so-called expression proteomics, it's also post-translation modifications. We have only less than 20,000 genes, but there is a lot of proteins in proteoforms, and they are here also because of post-translation modification. And actually we have a lot of proteins in our cell which are not functional unless they are modified, unless there is some phosphorylation, glycosylation, some other events which make them work. So there is on-off switch or some modulatory post-translation modifications which can switch on and off all those instruments inside itself. Also, there is a structural proteomics and proteogenomics, and also the emerging field is interactomics, how those proteins interact with themselves, with other proteins. Because actually, if you will try to name any not any, unfortunately, but most of the drugs which we use in medicine, what those drugs are targeted for? What those drugs modulate or prevents to work or are actually allowed to work?
Mostly protein.
Mostly proteins, right? And actually mostly three proteins: G-coupled protein, Y, some receptors, and well, two different labs actually. So, proteomics is really an emerging field. It's not some new field. It started like 30 years ago, but still, only now we have all the information to work with it correctly. And also, what we are interested in, and was interested 2 centuries ago, is where and when those proteins are expressed. So, also proteomics are often erroneously synonymous with any protein work, like someone is just producing some protein, cleaning it using chromatography and says that he is doing some protein work. No. Protein is a set of proteins. If you work with just one protein, it's a protein chemistry. But still. We have to characterize some proteins, and since we all humans, let's start with human proteome and see what is there is in it and what we can get from its study. And first of all, there is a lot of difference between protons of different cells. This article is available in our folder, and it states that even if we take 30 different samples from our organism, like a lung cell, some neural tissue, and so on, so on, so on, and just get those protons, proton of kidney, proton of heart, proton of blood, whatever, we'll see that if we'll try to get these numbers in such a barcode, it's a number of genes identified. So, of course, genes code for proteins, so we can count them as genes. And here is a number in what number of samples those proteins, the products of those genes, were found. And as you can see, there are some housekeeping genes and of course housekeeping proteins which needed to just have the cell alive. And on another end of the spectrum, we have a lot of proteins which, and genes as a products, which present only in one sample. So there are some characteristic protein for nervous tissue, for heart tissue, for whatever. So if we are studying proteome, we are mostly interested in those proteins which define our tissues or our cells, and not in those housekeeping cells, right? But let's say that we have our housekeeping genes and products of those housekeeping genes like FDH, tubulin, and so on, so on, so on, so on, including some histones and so on. You can look up in this article, which is also available. But if you look at this block here, are those housekeeping proteins at the same level in different tissues? No. Can we explain it by a genome or transcriptome?
No.
Not always. Let's say, yeah.
May I have a question? It is surprising for me that that according to the immunoblots, liver has the in comparison to some other cells, the lower level of housekeeping genes, because it is the hepatocytes are tetraploids, for example, because they have to produce a lot of, including housekeeping genes.
But why are they
expressed.
tetraploids? But why they are tetraploids? Why they have to have the genomic information in multiple copies?
I suppose because
It can be.
it should be more expressive. I mean that
Now, copies of genes not always directly compared to copies of mRNA or copies of proteins. Also, if I have some cells like liver cells, there is also some fatty inclusions, and if you will get this number of proteins per 100 grams, for instance, right, it wouldn't be the same as with, let's say, muscle, right? So it's not a direct comparison. And also, what I want to stress here is no difference between overall difference in protein quantity between those different tissues, but inside those tissues. So for aorta, for instance, there are two housekeeping genes which are almost not expressed, not, well, in English we can say that it's not expressed, in Russian of course not synthesized still, but there are two of them which are highly active, right? So usually what we are doing, we are trying to compare the quantity of genes of interest or mRNAs of interest or proteins of interest with a ratio to this host-keeping genes. But can we always do this according to this study? Right? So actually be careful when you are choosing which genes to use as a reference and which proteins. Yeah.
To be clear, could we compare liver, for example, with cerebrum, with the level of the expression of certain genes?
You can choose some.
So, for example, if I take first protein or housekeeping gene, could I say that the liver gene will not express so highly as its cerebrum gene?
You usually compare. Usually there is a ratio. You have this housekeeping gene, and you have a ratio of your gene of interest or a protein of interest compared to this gene. So here you see those housekeeping genes here in cerebellum expressed almost in the same quantity.
I talk about not comparing genes inside only one tissue, but between the tissues.
I see.
So, could we compare it or not? I understood that we should compare it in one tissue only, or it's not.
You can choose some housekeeping protein which is more or less stable across different tissues.
So it is stable.
But it's not a good idea to choose this housekeeping gene. I don't remember actually the first one. What was this? In aorta with, let's say, muscle, right?
Mhm. Yeah.
So not all classical housekeeping genes are good for comparison, but it's obvious actually. Let's get this GAPDH protein, which we use mostly as a housekeeping reference. Some tissues are more metabolically active, some are less metabolically active. Will they have the same number of protein? No. Also, if you already have had a transcriptomics course, you know that you don't get this raw counts of transcriptome. You do some normalization. What normalization have you done to your transcriptomes for these lectures?
We don't remember actually.
What do you usually do? You have an idea, an axiom, that there is the same level, total level of mRNA in your samples. And you say that you are counting and normalizing your genes of interest on your mRNAs in number of copies per million, for instance, and correct for the length of your transcript, right? But you still have this assumption that the number of mRNA in any cell is almost the same. The only difference is ratio to this mRNA, right? Is it always the case? So unfortunately, everything in biology, when we are talking about quantitative aspects is not as easy as it's in textbook. And answering your question, just see literature which housekeeping product is used for your area of research, and just use it and have some reference that you've chosen this housekeeping gene because of them. Blame them. It's they who says it is alright to do. Also, what is quite interesting, we are talking here about proteins, right? But here's differences between levels of mRNA and protein products. And as you can see, the correlation level between them is 0.27. Not quite much, right? So you can't absolutely predict the number of proteins in the cell having the number of mRNA in this cell. Why?
Because of folding, of protein folding, and
But
a lot of
why?
proteins can.
Folding is a certain structure of protein, and if there is some translation, you will get this protein product.
Yeah, we can say because we have this coefficient here. So,
But why we have it? Why there is no ratio, let's say 1 mRNA, 10 copies of protein products in itself? Why it's
not all
Different.
mRNA will be.
Are there any ways of regulation?
It depends on the needs of the cell.
It does, but okay. How the cell can get rid of some mRNA? Let's say that there is some mRNA which is not needed for this cell. Let's say there is some virus which invades this cell. How cell can actually get this virus mRNA off this cell?
Regulation.
Different types of RNA to silence this transcription.
Yes, mRNA infiltration, right. So even if you have those not fully processed mRNA in the nucleus, probably there is not any copy of this mRNA in the cytoplasm because it gets destroyed, right? And synthesis of proteins only occurs in cytoplasm. So it's one reason why it is so. Also, what are the other reasons? Are all mRNA have the same half-life inside the cell?
From the previous slide now.
Yes, you're absolutely right. It's not so. But why?
Poly A at the end.
Poly-A, right? But also, it's not only poly-A. You have processed mRNA. We have this poly-A here. We have a cap here. And also what we have, are there some regions which are not yet translated in the protein? Right? It's not that it starts from the beginning. There are some untranslated regions. And here also, right? There is some stop codon here, there is stop codon here, and only this part gets into a protein. But why we need those parts here, this part and this part in mature matrix, mature mRNA? Let's say that we have some eukaryotic cell which have naked RNA. What will happen to this naked RNA?
Yes.
There will be some protein which come here and, like a Pac-Man, get rid of it, right? And only if it's kept. And also it's actually this and this is like the structure. There is a cap, there is poly-A tail. And what will happen from time to this mature RNA? will it be degraded at once, or will something get shorter before it will be degrading? And only after some, right, number of this poly A, it will be get degraded. Also, are there any proteins which can decay our RNA?
It should be.
There definitely are. And what will happen to this mRNA after the cap will be stripped from it? The same, right? Also, what can regulate the number of events of translation from this RNA into protein? And also, what can prevent this degradation of poly tail apart from the number of this elements here? Let's start from the basics. Is it single-stranded or double-stranded? Single-stranded. And if it is single-stranded, will it be always in this linear form?
No.
Can it have some
Yes, yes.
structures? Can those structures actually direct this RNA to persist longer in a cell, or gets destructed easier by some enzyme, right? So those UTRs also play a role in this stability, right? So there's a lot of ways on how we can regulate either half-life of this mRNA or the efficacy of its translation in the protein. There could be just one ribosome. There could be a lot of different ribosomes sitting here and already doing their work, right? So this ratio of protein and mRNA is not one-to-one. Also, it can depend on a state of your cell, whether it is stressed, infected, developing, whatever. And this is actually what we're missing. We're only starting to get this information. System biology is what it's all about, this multi-omics approaches. So, okay.
What is the maximum on the graph?
1.0.
One.
It's a person. It's either 1, 0, or -1.
Yes.
As you can see, there is nothing at the minus part, so there is no anti-correlation at least. It's already a nice idea that there is no anti-correlation. Still, let's continue. What we're also missing is that there are some really rare events of mRNA editing. There is a lot of such events in plants, in animals, there is also such events, and if we are talking about us, humans, at least one event is documented in scientific literature. It's this apolipoprotein, which is, as you can see, big transcript with a lot of introns and extrons. And what's happening in some tissues? In liver tissues, it's the whole length, big protein with all the functions. But in intestinal cells, what we have is a special protein which can recognize this mRNA and get this. Just change one nucleotide. What is this nucleotide? What it will be translated into? Right? So you will get shorter proteins with the same mRNA. Can you derive it just from a genome? Will it be possible to derive it from transcriptome when you have some unprocessed mRNA, some fully processed mRNA, and it's something like 30% of U here and 70% of C here? What would you see? What do you think about such a result? Can it be that you have just two alleles of the same gene? Could it be just an error in sequencing?
Yes.
So you will just dismiss this as some error and so on, but it actually is. And if you have proteins, can you actually see that there is shorter protein in one tissue and longer protein in another tissue? So, as you can see, everything that you can imagine in regulation actually exist. So just have your creativity and just think about something impossible and then just search all the biological specimens and you will definitely find it. If it's not against the physics rules or something like this, it will be there.
If you cannot find it, try to find it.
Try
yourself,
harder.
yeah.
Try harder. Still, it is. So that's why we actually need our proteomics. And also one article which I cannot say that the results are valid, but I encourage you to read this article and find 3 points in their methodology which are, let's say, weak at least. It is published in Nature this year, but even for students, just read this article. There will be some points of concern. But still, this article is very interesting because this article states that so-called synonymous mutation, which have different codon but coding for the same amino acid, can, it's not always so, and they, in my opinion, haven't been able to prove it because of some methodological problems, but still can alter the total number of this protein in itself. Actually, from transcriptomic course, are there all the possible transport RNA in the cells for all the possible codons?
No.
No, right? And if we have some codon which doesn't have this transport RNA in cell, what will happen?
This RNA will not go to RNA.
It will be built. Actually,
Those
this
are not
wobbling
very
pair,
important.
this wobbling pairing, so it is possible, but will it be as efficient?
No.
No. So what they were trying to prove in this article, in Nature, that if we have a lot of such mutation, most of them will be bad. These budding yeasts will be will have lower rate of division. So for yeasts, of course, they have only one idea, is to proliferate as much as possible, and if they are not proliferating at the best speed that is possible, then it should be deterred. So also, can we get this from genome itself? Probably yes, if we have a really nicely annotated genome and we have all those tRNAs annotated and we know which of them are actually working, because there is a lot of pseudogenes which appears to be working, but just due to chromatin form or something else just don't get expressed. And the only way to do it is just to use lower levels of organization like transcriptome or proteome to see whether it's true or not. So, what was my point here is that we need proteomics, we need metabolomics, all the lower levels as well. But concerning this article, please check their methodological site. I don't know whether there are a lot of people discussing it, but on mobile.ru there was a flood about this article of different opinions considering the methodological part. Still, we need proteome. But how complex is it? What are the problems studying it? For genome, it's not only human per se. As you can see, there is different number of proteins, different number of genes, because, for instance, in plants there are more genes that provide us with protein products than in animals. Just different idea of genome organization. But still, the number is not so big. Transcriptome. Of course, we have different numbers because there is alternative splicing, right? And proteome. If we are talking about gene products, it's not as much. But if we include all the proteoforms, e.g. post-translation modifications, it could be a lot. Some sub-optimistic numbers, of course, nowadays some millions of proteoforms are discovered, but how many of them are real ones and how many of them are just some artifacts still needs to be discussed. And overall, it would be a bit lower if we are not including xenobiotics, but still big enough. So, some older publication, because there's a lot of genes for humans, which is not true as we know right now, but we have more transcript, even more proteins, and even more different post-translational modification of those proteins. So the complexity is bigger than in all the previous layers of organization. So one of the problems is that our protein is really complex. What are the problems? When we are talking about genomics and transcriptomics, we have only 4 building blocks, and they are quite similar. They have this sugar, they have this phosphate, they have this part which actually interacts and makes them pairs, and nothing more. So we can extract them easily just by using different solutions, and we can participate them, we can make them soluble again and clean and whatever. With proteins is not so, because we have at least 20 building blocks. If we add selenium and cysteine here, we'll have 21 blocks, and every one of them have different radical, different properties, different solubility, different, we'll be talking about this later, different properties. So there is no one way to extract all the proteins from your cell without losing some fraction of them. Also, the problem that we have here is high dynamic range. What we are talking about, that if we are talking about mRNA, there is logarithmic scale of copies inside one cell, and the red one is copies of the protein, and you can see that there is 100-fold, 1,000-fold even, a difference between them. It's one problem, and another problem that if we are, if we have some protein, let's say of human blood serum, the difference between the most abundant protein and the least abundant protein could be as high as 100 million. So you can have one copy of some really low abundant protein per milliliter of your sample and some tens of millions of albumin, for instance. And you have to measure them all to get the whole protein. It's like you have some way to weight bacteria from one side of your scales and, let's say, the whole mountain on other. And you have to actually be able to measure them at once. Impossible, right? So we will be talking about how we can deplete or enrich our sample for those proteins, to be able to compare something that is that have less dynamic range. Why is it so? Because mostly on one RNA there would be a lot of different ribosomes, and there would be more copies of just one copy. And if we'll try to look at a different graph from different publication, the same is still true. Also, the problem with our proteomics is that we can't easily get more copies of our protein. In transcriptomics, in genomics, we have polymerase chain reaction, and we can get from one copy to several million of copies and work with them without any problems, right? In proteomics, we have only one protein. We will not be able to get more, except for getting more sample in and using some clever ways to enrich your sample. If somebody would be able to provide proteomics with an equivalent of PCR, I think that a Nobel Prize is guaranteed. We will have easy time afterwards. Also, location and stage specific. We already told that a proton of different organs is different. We have seen that, but also localization inside the cell of different proteins is also different. And if you will try to get all the proteins of that cell, you will have to disrupt all the membranes, and you will have to somehow get all the proteins, whatever they will be, have more affinity to lipids, to water, will be charged or have no charge, whatever. It's impossible, but still. And there is such part of proteomics as localization proteomics, either at microscopic scale or at organ scale. Also, another example from plant science. You have a leaf. Let's say you have some wheat or rye plant and this long leaf, which seems to be just the same organ from its start to its tip. But if you just get this leaf and cut it in 7 different parts and get proteome from those parts, you will see that actually you couldn't fit all the data in one model. There would be some gradients, and for the tip of this leaf there would be one set of prevalent proteins. From the base, another. From the center is also another. So, what is the main idea here? That proton is really dynamic in both space, even for the same organ, like here, or time. And what we are actually trying to research is a snapshot of a part of this organ, no more. And the difference that we can find can be not the differences between, let's say, normal and diseased state, but just difference in sampling. Again, development stage. Of course, for such picturesque insects like insects with full metamorphosis, it's obvious that there is different proteomes. But even for us humans, even for the same organ, from, let's say, adult stage to some middle-aged adult, there would be differences. So it's not possible to say that this proteome is a final one. So, the last problem. We have a lot of data on transcriptomes, because it's easy to get nowadays. We have a lot of genomes, most of them not quite good annotated, but still. We have a lot of them. But for proteomes, if we are talking about reference proteomes, which are far from being complete, but at least at some stage of this development, nowadays it's only this much. So, a lot of work to do. What questions can be answered by using proteomic data? First of all, it's a phenotypic change in our system, and also it's possible that those phenotypic changes are not visible by naked eyes. So it wouldn't be getting you to the change in color, in weight, in length, and anything observable. But it can alter tolerance of your object for some given stress, for instance. And this change is hidden, so we can characterize this hidden phenotype, because why not include protein in a phenotyping? It's also a kind of phenotype. Also, we can actually just get two different situations, like developing with adult, like diseased with control group, and just compare what proteins are expressed at different levels, or what proteoforms actually expressed at different levels. Also, we can here directly get some insight on mechanism of cellular functions, because proteins are actually these machines which do the whole work, and if some machine is broken, we can just observe and see what happened to this system. So, functional proteomics is here. Interaction between proteins as well. we've already talked about that most of drugs are targeted for proteins, so we need it. And we can identify biomarkers, those proteins which are present in one condition or and absent or have different concentration in other condition. So that's what we can get with our proteomics. And of course there are some ways of describing different fields of proteomics. Protein mining, it's all about drug discovery, about screening those libraries of chemical agents against some protein, whether it can act as some stabilizer of it, or whether it can act as some ligand or whatever. It's also about finding targets for small molecules, and it's a differential display of proteins at different stages, different states. Protein expression is mostly about microbiology, cell transition, and so on. But what I want to stress here is that there are new additions as protein-protein interactions. Functional proteomics, we will be talking about it at our protein-protein interaction lecture, and structural proteomics. And since we've already characterized our topic of our lecture, proteomics, what it is and how it can be used, what methods we will be describing here, because proteomics itself doesn't have its own methods. It's either protein chemistry, bioinformatics, mass spectrometry, and we have to just talk about those methods. We'll be talking about electrophoresis. It will be one-dimensional and two-dimensional electrophoresis. We'll be talking about differential electrophoresis, when you can just directly compare two proteins without identification of proteins, but see other differences, and then you can get to identification. We will be discussing a lot of spectrometry-based methods, mass spectrometry, and some protein chemistry methods. And let's start with our basics, with our amino acids. There is 20 of them, and if we'll count serine and cysteine, it would be 21. If we'll count modification of like this one, it will be 22, but still 20, let's say, mostly common used amino acid. We have codons for, because as you know, for serine and cysteine we do not have a codon, right? How it is coded?
Randomly.
No. There is a lot of things that happens randomly in biology, but it's not the case.
So artificially.
No. Again, we have this single strand of RNA. Can there be some secondary structure?
Yes.
Is it possible that if there is some stop on the path of ribosome, it can signal for it to integrate some different amino acid, like serine and cysteine instead of cysteine? So, even if we have only 20 codons for amino acids, there are some ways to code for some different amino acids as well. And also we have these groups, amino groups and carboxyl groups, on almost every of amino acid except proline. Proline is a secondary amine, but it can still participate in peptide bond. We still can use it to lose water and have this covalent bond between proline and some next amino acid. Also, as you can see from the structure, there is a lot of different characteristics, because those radical groups are really different, and also all synthesized proteins are linear. they have this N terminus, C terminus, and if we get to some proteins which are not linear, then it's post-translation modification, some way to make them not linear, but they all start as linear polymers, getter polymers, of course. So, some terminology. We will have here a lot of names like dipeptide, peptide, oligopeptide, polypeptide, protein. There is no strict definition, like 10 amino acids is peptide, more than 10 amino acids is polypeptide, but just some rule of thumb, anything that is less than 50 amino acids, but more than 2 amino acids is a peptide. Something from 2 to 15 is oligopeptide, something from 15 to 50 is polypeptide, and anything that is more than this is proteins. And don't get me wrong, but proteomics also work with peptides, and some functional proteins which synthesize as big proteins are modified to shorter proteins which have less than 50 amino acids, but which are considered to be peptides, but still proteomics is used to get them. And talking about such an example of a small protein or peptide, if you want, is something that was first sequenced protein, insulin. And it was sequenced by the guy you already know by sequencing of nucleic acid, by Friedrich Sanger. Actually have 2 different Nobel Prizes, and his first Nobel Prize was for sequencing the insulin. So he have only 2 publications in more than 10 years and 2 Nobel Prizes. Still, if we'll try to see the structure of the insulin, you'll see that it's not quite a linear peptide or lipoprotein. There are 2 polypeptides here with some covalent bonds as a cysteine. Cysteine is really interesting amino acid because, let's see here, it have sulfur in it, SH2, and what will happen if 2 SH2 groups will be close enough in some reducing conditions?
Be sulfur rich.
Absolutely, yeah. We will have covalent bond here.
Yeah.
So only cysteines are capable of such kind of covalent modification, and you see it here. There are at least 3 of them. How this? Let's a bit further to talk about. I just missed. Yeah, I just missed the order of slides. So sorry. How this protein can get to this structure? Actually, it is produced as one long protein, and it has some different parts, like signal peptide, which is produced first, and this signal peptide stops this process before it is docked to endoplasmic reticulum. So only after it is docked on this endoplasmic reticulum, then the remaining amino acid can be added. Then we have this protein, and we have the first case of protease events. Protease is a protein which cleaves other proteins at some specific locations. There are of course some proteases which can cleave almost everywhere, like succinylation for instance, and there are some really specialized proteases which can make this cut at really set sequence of protein. And the first is cutting here. Since there is differences in pH inside and outside in the plasmatic reticulum ribbon, there will be self-catalyzed forming of these cysteine bonds, and we have this form. And after it is exported in Golgi apparatus, we'll have second round of different proteins which cleaves here and here. And afterwards, there is a third event of cleaving those two amino acids. We get our insulin. So, we already have seen a lot of post-translational modification. Since post-translational modification is covalent modification of proteins, can this event of protease activity be considered one? Why not? We broke some covalent bond, right? Our product is not the same as the gene that coded for it, right? So we post-translation line, translation is already occurred, modified it, right? And what do you think? Other ways to have this proteases in prokaryotic cells? Are there any protein in bacteria which acts as a protease?
In bacteria, proteins which act like proteases?
Yeah,
Or
of course. And there is one common misconception, that there is no post-translational modifications in bacteria. But we have one here, right? So it's a misconception that post-translational modification cannot happen in prokaryotes.
I believe that glycalization can be.
Absolutely. And glycalization as well. But in some old books there is a statement that protein post-translational modification are a feature of a eukaryotic cell and so on. Not the case anymore. Maybe I'm just old enough and I remember it, but still. So, as we can already see, we've got this linear protein here. And of course there is more than one level of organization of proteins. Of course we have primary structure, just as sequence we have secondary structure, those beta sheets and alpha helixes. We have tertiary structure when those alpha helixes and beta sheet position itself in a certain way. We have quaternary structure, we have some protein which consists of different monomers, they can act. Actually, there's only two classifications of those structures. It's either globular protein, which is like this, or filamentous protein like actin and so on. But let's start with this secondary structure. As you can see, this structure is mostly because of those hydrogen bonds. And as you can see, radicals of our amino acids are outside these alpha helixes. They are exposed. And if we are talking about beta sheets, we have the same hydrogen bonds here, though they have a different direction. And radicals can be either on this or this side. And why this is important? That most of our proteins have both of those secondary structures and have some position of the structure, we need to discuss why this folding can happen, what are driving forces of this folding, and why some proteins can be folded even by themselves without any help. And other proteins need so-called chaperones, proteins which helps them to fold. What do you think concerning our radical groups which have different properties? What can lead to this spontaneous naturation of this protein? How can it fold itself in some ways?
Positive and negative charge, for
Right.
example, and they
First, absolutely right.
Hydrophobic on
Hydrophobic and hydrophilic, right.
Or gives energy.
Yeah, absolutely. So there's a lot of ways how this can be possible. But for most of proteins, what do you think? Is there only one way to fold this protein?
no.
Of course, there is different ways how it can be folded, but protein will try to have minimum free energy. And if it is possible to do, it will fold in this way. If it's not possible, it will be some different. But also, what do you think? Will it be true that this form of minimal energy would be the same at different pH, temperature, different salinity, and so on? So it's also a factor of how you can get your protein either naturated or denaturated. Just a school book example, when you boil egg, you have this denaturation of protein, right? So, first idea is that if we have some hydrophilic and hydrophobic groups, it's absolutely possible that those hydrophobic groups will try to clump together and form something inside your protein, and all the hydrophilic groups will be on the outside. Mostly it is the case, for instance, green fluorescent protein, right? There is this barrel, which is absolutely hydrophobic, and why this protein can fluoresce? Because this active fluorophore is in a water-less environment. If you have something that will crack open your GFP, it won't be fluorescent anymore. So most of the proteins actually try to get this shape. Also, we have hydrogen bond, this beta sheet structures, we have hydrophobic, hydrophilic interaction, this water-free pockets, we have salt bridges. As you can see, there is positive and negative charge, and we will try to attract each other. But if there is the same charge, it will try to repel us. Also, driving force. We'll have this disulfide bonds, which we already discussed, and it's mostly the repertoire which will define how a protein will try to form. But there is a lot of ways how the protein can fold, and before the break, the last slide, is that if we'll calculate this free energy for different ways of folding, we'll see something like this. It will have some landscape of folding. Unfolded protein will have a lot of free energy. It will try to somehow get rid of it. But there are some local minima of those free energy. And some local minima could be the real one, the native state of our protein. But it's possible that it's not the lowest of possible ways. And some proteins can spontaneously reiterate. For instance, our famous GFP. It can reiterate after boiling without any problem. But most of the proteins need some helpers, some chaperones. And why we have so much of heat shock proteins in our cells? Because if we'll heat up a protein, what will happen with this energy?
Growth. Energy growth.
Which energy grows?
May you repeat the question?
Yes. Why proteins will denaturate at higher temperatures? This is still true, but can it get rid of this energy if there is a high temperature?
High temperature means that atoms have more energy.
Yes, and this is still
I
true,
don't
but the energy is the same. Why should then it fold if it will not get the lower energy for it? If the energy of the whole water and cell is higher than the gain that this molecule can gain by just folding, right? So if we have some higher temperature, our proteins can denaturate, and we need those chaperones which help them reintegrate in a right way, because there are such local minima which can get out either to amorphous aggregates or some amyloid fibrils. For some proteins, not all proteins are able to get to this form, but still. And what chaperones usually do, they prevent moving from this local minima to those local minima, and tries to get some energy to get over this hill to this low energy state. So I think that it's all for this part of lecture. If you have any questions, you can ask. And we have 10 minutes break.
Can we open the thing?
I think so. Let's check if it is. Yeah, it's possible.
Приходите.
Хотела спросить маленькое уточнение.
Да, конечно.
По-моему, вы даже сделали ремарку об этом, но вначале я записала. Просто можем ли мы говорить об экспрессии?
Смотрите, экспрессия по-русски, только у генов.
Ну вот да, я поэтому...
По-русски белков синтез, но в английском и то без
То и
expression.
то.
Учитывая, что мы перешли в разряд кальки с английского, раньше за это очень сильно наказывали. Сейчас вроде как уже несколько защиток слышал даже на кандидатских, когда говорили о экспрессии белков, вроде как это уже проходило без особой аллергической реакции.
Мне стало то, что непонятно, но я
Да, конечно.
не могу понять в своих записях, что мне осталось непонятно.
Но, в
Они
принципе,
просто у меня
вряд ли
тут тезисы, поэтому мне есть вопрос, но мне нужно вспомнить его.
готовьте вопрос.
По-моему, это было что-то связанное с...
Может, слайды.
Да, может
Если
быть.
есть номер слайда, скажите, перелистну.
Сейчас, он же есть на этом?
Да, не папки.
Я посмотрю.
Конечно. Слушайте, пока по-русски вопрос нужно так подробно или можно без этих введений?
Мне нравится подробно. Ну, если это возможно.
Хорошо, просто мне кажется, что я это все говорил, потому что это несколько лет уже подряд. И каждый раз, когда читаешь, возникает такой дежавю.
Visualization. So now we're going to do this small. That's not what and now we have different specifications, so maybe we have different assumptions.
But now they ask us to merge into dominant specialization. I am not in the same specialization as previously. They told us to change because they were closed.
So now
А, ну,
And then I know it's
yes. I see, yeah. So
no
you chose
programs.
yeah, I see. So maybe some people are home, see.
Я немножко пропустила. А как у нас будет проходить зачет?
Значит, у нас будет зачет по двум сразу частям. Первое, это нужно будет сделать доклад по темам, там будет свободная тема в том числе. Я, по-моему, выложил уже в папку курса, там, в очных материалах тему докладов. Можете предложить свою, если вы хотите, но я могу это согласовать. И будет онлайн-тест в Google формах. Там, 50 вопросов у меня было в прошлом году. В этом году 53 вопроса. Вот, и будет набрать количество баллов. На этот тест будет больше дня, потому что там, в принципе, тест не столько на напоминание, сколько на то, чтобы вы смогли найти, посмотреть.
А, ну то есть это вот не по времени. Просто,
День.
ну нет, в смысле, ну не 1,5 часа. Просто у
Слушайте,
меня получается,
смысл в магистратуре спрашивать
не знаю. Просто у меня сейчас получается летняя школа еще параллельно идет, и поэтому я буду опаздывать. Она там обычно до 6-7 часов.
Хорошо, лекции в любом случае все выложены в ДТ, так что можно будет посмотреть, что спрашиваете. Если все согласятся, можно начинать чуть попозже, например, если нужно.
Да, я сомневаюсь, что кто-то согласится начинать из-за меня чуть-чуть попозже. Но у меня все равно там не каждый день до... Да.
Мне вот тут еще запись веду просить, так что, в принципе, будет
Да,
запись,
да, у нас же
можно
есть.
будет с неё.
Все хорошо, все.
Я помню, что это связано с селеноцистеином. Кажется, я не поняла, наверное, просто сам принцип его
селеноцистеин.
синтеза. И я в какой-то момент поняла, но не поняла момент. А потом я забыла, что я поняла. Потому что я как-то...
Селеноцистеин и селеноцистеин у нас кодируется одинаково, кодон тот же самый, да?
Да.
Но так как у нас может, в принципе, особенно этот прокривод, образовываться шпилька, рибосом на шпильке тормозится.
И он как бы
Если
добавляет
тормозится,
дополнительную
тогда да.
аминокислоту. Просто я не понимаю, откуда там появляется еще...
Ну, просто под него нету самого кодона.
Сейчас я хочу
А
посмотреть.
на Википедии, по-моему, есть. В английской точно, в русском не помню. В английской Википедии точно селеноцистеин, там у нас картинка.
Просто откуда там берется селен?
Ну, это только ленинских ферментов нужно. И на самом деле, если вы витамины посмотрите, которые мы даже едим, там есть селен.
Я скорее вот в процессе синтеза белка, просто как кофактор какого-то фермента, не знаю, не то, что кофактор именно в ферменте выступает, а просто именно присоединяется в момент синтеза.
Нет, не в момент синтеза.
То есть это можно считать
Там
присоединение как тоже
такая,
последовательная.
такая есть. И с одинаковым, соответственно, кодоном. Но одна из них не очень подходит, вторая подходит больше. Давайте на следующий раз просто попробую анимацию найти в YouTube, чтобы показать.
Я поняла, что мой вопрос странный. Они же берут просто уже готовые селеноцистеины. Они не происходит же синтеза селеноцистеина в нашем организме.
В нашем нет.
Все, я просто у меня...
В бактериях происходит.
Да. Ну и просто он уже синтезируется, у меня
В
чувство,
наших
что
организмах вообще очень много есть аминокислот, которые нами синтезируются.
Да, я помню. Просто у меня иногда как-то
Но,
путают в голове.
к сожалению, некоторым существам их можно сами производить. Например, растениям, откуда их взять.
Ну да, да.
Ну, конечно, есть микробиом, но опять же, микробиом тоже можно синтезировать. Так что определенных видов, скорее всего, прокариот, все-таки есть фермент, который позволяет их синтезировать.
Да, я поняла, что мой вопрос про откуда селен, просто он немножко из другой области. Я подумала, что он
не синтезируется.
что изначально цистеин туда добавляется, и потом происходит модификация какая-то. А именно, да, она немного неправильно
Просто
у меня в голове.
каким образом, не имея кодона специфичного, можно заменить. Вот один из вариантов.
Да, наверное, так. Вот, короче, вот так же что было. Не потому что ты ребята. Супер,
Я пришла, послушала Сашу. Я хотела спать, но слушала Сашу. Вот Марина голодная. Когда?
Как активировать здесь на описании? Не хотите с нами сходить получше?
Я думаю, после.
Ну, после.
Я поставил еще электричка до Петергофа, нужно попасть, поэтому
Ага.
тянуть будет со временем.
Ага. Я только кушать.
Так, вот здесь хорошая кафешка, там кофе очень вкусный.
Здесь?
Кофе вкусный, да, при входе.
А, да. Я подумал.
А там еще на 5 углах вон происходит, у них новые носи.
А,
Уже можно попробовать.
не, я удивился, что в институтах бывает хороший кофе.
Тут везде, тут слева, справа кофейни, там сбоку, в какую сторону мне пойти.
Кофе недавно, то, что хорошие, вот это удивительно. Это было, да. Вот. Теперь каждый раз, когда спрашивают, как было в ИТМО, ты еще рассказываешь, приходит вкусный кофе. Вкусный кофе.
А вы у нас круглую преподаете? А политех?
В политехе, ну, в этом году какой-то курс есть, просто какой-то был, да, да, был. Right, let's continue then. I think that this time is already. I think it would be better that we will just end a little bit earlier, right? And have a bit shorter free time between lectures. So, we have discussed the problem of folding of proteins, and I want to ask you some control questions about it. And the first question, would the protein of the same amino acids sequence and same lengths fold in the same way always? No. If it can fold in the same way and different pH, temperature and so on, will it fold in the same way in different temperature, pH?
Not always.
Not always, right, absolutely. You have a chain of amino acids. Some of them are polar, some of them are non-polar. And you just synthesize this protein. Is it possible for this protein to have some folding by itself? And what will result in this folding? Which residues will be outside and which residues will be inside this protein?
Polar outside, non-polar inside.
Which more readily deal with water? Polar or non-polar?
Polar. No.
Polar or non-polar? Fat is polar or non-polar?
Non-polar.
Non-polar. It doesn't deal well with water, right?
Therefore, I said non-polar inside and
Non-polar
polar
inside.
outside.
Polar outside, right?
Yes.
But no, but
You said the reverse.
I heard that it was the other way around.
Yes.
My
So
people
it's-
think,
Okay.
I'm sorry, in my mind it was like polar outside.
First day of new semester, it's always wrong. So, right. Then, what do you think? Which protein will have better solubility? The protein which have some different polar amino acid that are randomly get through the whole protein, or protein which have some part of it covered with polar amino acids and some other part with non-polar. Which would be more soluble?
Polar
Second one.
second one.
'Cause
What
there,
will it be actually?
there is a structure, maybe. Yeah. Maybe both of them.
No, well, it depends, but mostly, if you have some small number of polar amino acids which are randomly occurring in your sequence, that it would be soluble more or less. But if there is a big region of polar amino acids and then there is a part of non-polar amino acids, this polar head wouldn't be soluble in water, and it will try to aggregate with the same protein, or will try to just somehow get into membrane, right? Where it will be absolutely happy in this polar environment. All right. Actually, just one question which I haven't discussed yet. Is it possible that some proteins require some post-translation modification for correct folding? Could it be so? Or another way, is it possible that protein will not be able to correctly fold without some post-translational modifications?
Yeah.
No, we have, I don't know, pH and temperature is it modification?
But let's say
When we are talking about modification,
Yeah.
when we cleave part of some protein, so it looks obvious that the folding will be different.
Right. So for some proteins to be correctly processed and to be correctly folded, there is a need for some certain post-translation modification. If those modifications are absent or some different modifications are here, then it can be misfolded. For instance, there is such protein which have its role in progression of Alzheimer's disease, tau protein, and when it have two phosphorylation sites with phosphate, it can clump together, forming those neotunnels and so on. So it also have its impact on protein folding. Then we've talked about the folding of proteins, but also most proteins do not work by themselves. They either have some monomeric subunits which need to get to some certain form before this protein can function, or even more, some proteins need to have protein-protein interaction to have some protein complex to get it all done. Usually this quaternary structure is illustrated by hemoglobin, but it's a textbook example, not quite interesting. Let's take example from neurobiology. As you know, many of receptor and many of ion channels in our nervous system consist of some monomers, so it's either pentameric or hexameric, and it's not quite seldom, or I would say on the contrary, it's quite frequently that those monomers are a little bit different, and for some ion channel or for some receptor to work, there is a need for different subunits to be present in this complex. For instance, there is such a channel which provides chloride transportation to the cell and out of the cell, and it is also called as gamma aminobutyric acid A-type receptor, this GABA A type of receptor, which consists of, as you can see, 1, 2, 3, 4, 5 different subunits. And it is functional only after those subunits are at the same place and they form this big protein. And what is interesting is that there is different types of this GABA A receptor. If different subunits and different numbers were used to create this receptor. For instance, there is such types, and as you can see, they both are quite like. But in one case, this receptor, if we'll activate it, provides a sedation of a person or animal. In other case, it will have some anxiety, and of course, anxiety is not a good thing to have. And if you have some small drug or something that interact with this given receptor or ion channel, you would like to have this sedation effect, but have no anxiety effect, right? If you just want to get some drug on a market which you can call sedative drug, it would be not quite nice to have anxiety with a sedation effect. right? And it happens that this difference in the subunit organization can dramatically change where this receptor is and what is this receptor's characteristics. So this quaternary structure or level of organization is quite important. And also you know about this drug called diazepam, and as you can see you have different subunits. In some cases that have no effect at all. And there are some mutations in our population, or where some person lacking just one of the subunits and provides with this type of receptor, and diazepam doesn't work for them at all. And also some anxiety disorders are linked to some differences in this subunit organization of this receptor. I think this example is a little bit more interesting than hemoglobin, which is mostly in textbook, because it provides function to this different organization. Also, there is one more level. It is not coined yet in the textbook as a fifth level or a sixth quaternary sixth. I don't know how to say fifth level using Latin naming, but still fifth level organization. Is the work of big protein complexes. I think that you've already have some course on molecular biology, and you know about this bacterial nucleosome organization. So when a bacteria tries to replicate, usually all the proteins that needed for this process are tightly packed together. And the central protein here is this clam loader, like a little squid here, which actually orchestrate all this process. Of course, in eukaryotes we have the same process, but we don't have this clam loader, at least in this form, and we don't have this all proteins interacting so closely, but we can actually speculate why is it so. What would be for what reason we would want to have all the proteins needed for replication in one place? What can be beneficial if we have all
reaction.
Right. For bacteria, it's a pinnacle of bacteria to get most copies in less time, right? For eukaryotes, we have some other things to do, not only just multiply our cells. And we have some regulation. And if we have all these polymerases, helicases and so on just floating in some near our nucleic acid, it's enough because on such small distances, even free diffusion is fast enough to have this reaction gone. But for bacteria, even if you're 1% faster than other bacteria, you will survive, proliferate, and take over the place. So that's why, for some cases, we have such big complexes. Let's say fifth level of organization, right? Because helicase is of monomers, it's our quaternary level of organization, and we have, you see this clamp loader, 3 polymerase, beta clamps, and also there is primase liking. It also have some affinity to helicase, so it's a multi protein complex, which achieves high local concentration of this protein in this place. So actually what we do, we have high concentration of this protein in a small volume, but we can have low concentration of this protein elsewhere, right? So those protein machinery, those protein complexes actually needs only for one reason, to have high concentration in small space. And they all actually not only stay there together, they actually have some ways of interact. For instance, if there is some problem with replication and polymerase is lagging, you will have this trombone scheme when there's there will be a lot of single-stranded DNA here, it will inhibit our helicase. So this complex will just stop moving further unless this single-stranded DNA will be processed. So it's not only because of local concentration, which is important, but also because those proteins can interact with themselves and with other proteins. So, as you can see, it's a really interesting question, and of course, there is no defined 5/5 level of organization of protein, but you can see here, we can think that, well, if we have no such abbreviation and no such term, still it's here. Then, actually, we have a lot of such complexes in some place, at least in eukaryotic cells, because of compartmentalization. Because eukaryotic cells can have membrane inside their cells. For instance, synaptic vesicle. So it's a small lipid sac with some mostly primary amines, which are mediators in our central nervous system in it. But we need a lot of different machinery to have low pH inside this vesicle, for docking of this vesicle, for releasing of this vesicle, for transporting our, let's say, it's glutamate, I think we do it here, yeah, to transport glutamate in it, and so on, so on, so on. And they are all actually have to be in this vesicle for it to function. And as you can see, it is a reconstruction. The article is also provided. It's a reconstruction from cryo-electron microscopy, and you can see that there is, I think, a lot more proteins than lipids even in this vesicle. So whether it's a vesicle with some proteins or proteins with some lipids, it's up to you to decide. So it could be our 6th level of organization. Not all proteins, but still. Now, because we already know that those radical groups have a lot of impact of how protein is folded, of how protein is functioning, we have to group them by some characteristics, and some of them are hydropathy, hydrophilicity, hydrophobia, and Alzheimer probably, hydrophobia, hydrophilicity, and as you can see, there are highly hydrophobic amino acids here. There are hydrophilic, and there are cysteine, histidine, and methionine, which is moderately hydrophilic or hydrophobic if you want. So we already can group our amino acids just by this characteristic. Why we need it? Proteome is quite complex, right? And if we have some ways to simplify it, to analyze all the protein not in one time, but somehow get hydrophobic protein, moderately hydrophobic protein, hydrophilic protein, we already have 3 different fractions, and each fraction will be 1/3 less complex, right? Also, we have charge here, and most of our amino acids are neutral. Of course, we are talking about amino acids which already have a peptide bond to other amino acids, because if we take in consideration COH and NH4 group, there would be some charge, right? So we are talking about only radical group here. Most of them are neutral, but some neutral with a green color here, because at some pH those groups can have charge, but it's not physiological pH. It can happen that this pH can be achieved in lysosomes or some other condition, but mostly in cells they are neutral. Also, we have positive 3 of them and negative 2 of them charge, and since those amino acids have a charge, we can actually try to separate them or proteins with them by electric charge, right? And it will also provide us with a way to simplify our mixture. If we can, let's say, have 10 different fractions of a whole protein just on this native charge, each fraction will be 1/10 of the problem. It will be easier to analyze. And also we have such characteristic as solubility. As you can see, there are some amino acids like tyrosine which are almost insoluble in water, and some of them, like lysine, are easily soluble in water. So if we can just have different concentration of salt, let's say ammonium chloride for instance, we can participate our mixture by having some of our proteins in solution, while other at this salinity are already participate at the bottom of our epidorf, for instance. Right? So we have another way to simplify our complex protein. Just if some 30 years ago there was one person from our military academy who says on one conference, just give me an ultracentrifuge and a bag of ammonium chloride and I will simplify all your protein down to just some proteins. Of course it's not so, but it's possible to do so. It's possible to simplify just by using different salinity to participate some of protein. But what we are most interested in today is this pK of radical growth. It is here. Since we have different charge group on our amino acids, let's start with free amino acids. It will be easier to understand, and then we will get to protein level. As you can see, at different pH, we have different charges here. For instance, if we have alanine and we have acidic pH, we'll have positive charge on this group. Why this is happening?
What's happening?
This charge, why
This
is
charge
it?
because of hydrogen.
Yes, because of
bounded
hydrogen
to
and inorganic chemistry, buffer solutions, right?
Mhm.
And if you just change pH at some basic site, let's say more than 10, there will be no positive charge on this group, but our carboxyl group will have this negative charge, right? And in some intermediate pH, let it be pH of 6, we'll have two of those charges on the same amino acid, right? So if you know this pi of each group, we can find the pH at which this particular free amino acid will have no charge. And let's talk about such a strange way of electrophoresis as electrophoresis of free amino acids. Let's say that we have a gel or solution, doesn't matter, with pH 6, and we have 4 different amino acids. Alanine, which have pi of about 6, isoleucine, which is also about 6, arginine, which have about 10, and aspartic acid, which have about 3. And we start with putting them at the center of this electrophoresis cell, and then apply some voltage. And as you can see, at this pH of 6, we'll have our arginine moving to solution to cathode and our aspartic acid to anode. As you can see, we already can separate some of our amino acids by this simple electrophoresis. But what if we change pH, let's say to 10 0.8. What will happen? We have the same electrophoresis cell, but the pH of our gel or our liquid will be different. Let's say 10. What will be the movement of those free amino acids?
All goes to the plus,
Except
except this one here.
this will be
Will
in the
stay.
central state, and if we'll have this 2 point, well, let's be 3, then it will be vice versa. Those 3 will move here, and this will stay, right? So, what we have here is we can find so-called isoelectric point of our amino acid, at which pH this amino acid will have no net charge. We'll need it later for 2D electrophoresis. And with free amino acids it's quite easy to do, and actually if you need to find this pi of iso point, you can just sum those pK of those 2 groups together and just divide it by 2. We have pKa of this group of 9.69 is 2.34. We can add this, and if we divide it by 2, we'll get about this value. So for free amino acids, it's quite easy actually to get this number just by simple mathematical rules. Fortunately for us, most of those pKa's for both groups are calculated, as well as pK for our radical groups. Because for some amino acids, we have additional charge on our radical group, and we also have to take it into consideration. For instance, let's try to see it for arginine. For arginine, we'll have not only 3 different variants, we have only positive, only negative, and positive and negative charges, but we'll have 3 different charges, and it will differ on pH. When pH is acidic, we'll have 2 positive charge. When our pH is highly basic, more than 13, we'll have just 1 negative charge. When it is about physiological condition, we'll have 2 positive charge and 1 negative charge. And we will have pH of basic about 11, we'll have 1 negative, 1 positive charge. So it is understandable that for this pH, we'll have no net charge on this molecule, and it would be an isoelectric point of this free amino acid, right? How can we calculate it? We need to know which of them are closer to this state of having no charge. And then we have those 2 numbers. Again, we need to sum them up and divide by 2. And we'll get the isoelectric point of this free amino acid. Was it understandable about free amino acids? Then let's continue with proteins. With proteins, we have this peptide bond, and there is only 1 positive and negative charge on N and C terminus. But we have a lot of the different radicals, and some of them can also have charge at different pH. For instance, we have this protein, we have cysteine, we have lysines, we have some other amino acids, and for highly acidic pH, we have 1, 2, 3 positive charges, so it has 3 plus charge. For some more basic pH, we have 2, 3 positive and 1 negative, and 2 negative. And some other pH, we have 2 positive and 2 negative. And for this pH, this polypeptide will have no net charge. And if we'll get to some high pH values, we'll get a lot more negative charges. And as you can see, that even cysteine will have now negative charge. If you remember, I marked those cysteine and tyrosine as negative at physiological pH, but if you have really big pH of let's say more than 8 and more than 10 here, it can get its charge. So you can see it's not physiological but possible to have. And if we can calculate it, and it is possible to do using some online calculators, it's not needed to do it by hand, we have another way to separate our proteins. We can actually have electrophoresis at certain condition, at certain pH, and only proteins which will have no net charge will stay in the solution. And once we have positive or negative charge, we'll move to cathode or anode respectively. So it is also possible to do. Let's see if I have some ways. There was a link somewhere. Just 1 second. Peptide calculator should be here somewhere.
Previous.
It is here. So yeah. We have a lot of different websites which allow us to calculate these characteristics. And for instance, we can, let me make it a little bigger, put here our sequence. Let it be some protein from plants. We can select modification of an acid terminus to get the real pH. We can actually say which disulfide bonds are there, but there is no disulfide bonds here, and we can calculate easy all that we need. It is possible to get molecular weight. It's possible to get isoelectric point. As you can see, it's slightly acidic for this protein. We have the same graph here, and also we can get solubility, whether this protein probably would be soluble or insoluble in water. So you can play with this peptide calculator to see how different amino acid composition and sequence could change the property. So if we want this peptide from our mixture, we can use this pH and electrophoresis, and this peptide will stay in the solution, and all others will have some different charge and will move either to a cathode or to anode. So use this calculator if you need those characteristics. Why it is of value for us? As I've already said, the problem we are facing is that proteome is really complex, and to have a way to work with such a complex proteome, we need to separate it. And one of the way of separating it is using a so-called isoelectric focusing. I will try to describe this apparatus, how it is working, but I will need some whiteboard here. Let's say that we can actually put in our solution some chemicals called amphibolites, which themselves have some charge and some pH. We have, let's say, negative and positive plates, and we have some amphibolites, which, if we put some current to our cell, will position themselves according to it somewhere in this cell. But those amphibolites also have groups which will have some pH. So if we'll just run this cell, and at some point it will separate in this way, and measure pH in this cell in different regions, we'll see that there is a gradient of pH forming in this cell, along with the gradient of electric field. Right? But amphalites is not our proteins. It's not what we are interested in. But during this run, we already get two different things. We get a gradient in pH, and we have some gradient in voltage in this cell. And if we'll put here our proteins, what do you think? What will happen? Let's say we have one protein, let it be blank big protein, which have some pH of, let's say, 4.1. We have pH gradient, and somewhere here, let's say here, we have pH of 4, because there's amphalites which are here, which have a lot of groups from this pH. What would this protein do in these conditions? If this pH, where he is let's say 6, and it's his pi, it will have negative charge. Where will this protein try to move? And what will happen to this protein when it gets to 4.1 of pH?
It becomes more
Positive. If here, let's say 3, it will get positive. But what will happen with it right here?
With the nature.
No, the nature actually, it's not a harsh conditions. Will it have any charge in this position?
No.
Will it try to move anywhere from this position? If it will move by just shear law, by some just diffusion here, what will happen? It will try to return position. If it will move here, it will try to return this position either, right? So what we have done here is so-called isoelectric focusing. We use negative charge of this protein to actually separate it from other proteins with position. At this pH, this protein will not move anywhere. So if we'll just have our proteome and put it in this cell with all our hundreds or thousands of proteins, they will all align and they given EI, right? And if you try to move somewhere, it will be returned to its position. And then it is possible to have this vessel which have semi-permeable membrane. This have big pores, so proteins can easily diffuse from one vessel to another, but we can then collect simultaneously water from each vessel to our Eppendorf tube. And what we will end up with, we will have our protein mix simplified. Here we'll have pH from 3 to 4, 5 to 6, and so on, so on, so on, so on. Right? So, just this simple characteristic of amino acids already provides us with 4 different ways of separating our proteins. Charge, solubility, hydrophobicity, hydrophilicity, and the size of the proteins, right? So we have at least 4 different ways of simplify and get information of our protein. If you want to see more about this machine, there's a YouTube link. It's possible to see how it works. They actually demonstrated on some proteins which are natively colored, some cytochrome and so on and so on. So it will be really nice to see how this brown mixture gets separated by red, green, blue and brown stripes in this machine. So, just to sum up, we have different characteristics of our amino acids, and they directly allow us to simplify our proteins just using these characteristics. At least 4 of them are usable: size, hydrophobicity, hydrophilicity, charge, and solubility. Also, you know that not all of those amino acids are synthesized by eukaryotic organisms. Most of them are not synthesizable, at least by humans, and we need to get them from food. And unfortunate thing here is if we'll change our diet, not all of those amino acids will be accessible for us. For instance, I know there's a lot of young people who tries to get to vegan type of diet, but some of those amino acids are not available at needed quantities. So if you will go into vegan type, please use supplementary addition of needed amino acids. It's a good way to have right diet. Also, what we have here is amino acids which are actually 20 of them, which have some codons for them. We already talked about Pavel Zykin and acetylcysteine, which do not have specialized codon there, but could be also incorporated in our proteins. The question is actually just to have our imagination work. From chemistry point of view, what do you think? Are there any other amino acids except for those 21 or 22? And why only those amino acids are used by living organisms?
There are more than 22.
Absolutely.
Why are they used?
Personally, that amino acids are in cells.
But why? Why? Actually, there is a lot of amino acids that are in cells but not used to build proteins. Ornithine, for instance, gamma-aminobutyric acid, for instance, and there is a lot of different primary amines in our cells, and we even have receptors for these primary amines in our cells. It's strange that we have such a receptor in our olfactory, in our noses, and our immune system, and our brains.
Just because.
It's a good answer actually, just because it can be in biology, because evolution doesn't answer any questions. It just happens to say it happened.
It works like that.
It works like that, yes, but in different conditions probably. If we are talking about nucleic acids, it's easy to actually have some ideas why we have those nucleotides. Because they are more stable, they are not as prone to degradation by ultraviolet light, and so on, so on, so on. So from primary bouillon of different organic chemistry that was on Earth, they were most stable, they were winners on this race. But there are some different nucleic acids that are used actually by living organisms, if we can call viruses a living organism. But yeah.
Maybe those amino acids just break in proteins somehow and not making them work properly.
But we actually need some acetylcysteine in some cases. It's a question that actually we don't have one textbook answer to this question. I'm only asking it to make you not take everything that is taught in this course as pure truth. You know, what I'm talking about today is truth for today, but tomorrow there will be different ideas, there will be different researches, and probably something that we today think as ground truth wouldn't be anymore, right? Like nobody could even think about RNA editing events in living cells like 10 or 15 years ago. Nowadays it's obvious that there is so. So just try to think for yourself why it is so, why those amino acids are there and so other. Actually, what is also interesting, why are we using only one chiral isomer of amino acids and only one chiral isomer of sugars in biology?
Just because I remember.
No, no. It seems that if we'll try to get some information from comets and from other bodies in space, that it is possible to form such complex molecules just by chance. And somehow molecules with such chirality is more stable under ultraviolet light. I can explain it. For me, whether it's right or left, why it is so, but there are some works that actually shows that actually Japanese space agency gets some data from Aribo, if I'm not mistaken, asteroid, and they were able to find some traces of amino acids and sugars, and also the same chirality as we are using here. So let's continue. Amino acids have properties such as size of radical group, such as charge, such as hydrophobicity and specific relativity, and proteins have just a number of these building blocks. It's more than 4, it's not some DNA or RNA, have a lot more of this, and we can use those properties either to work with our proteins, or proteins use those properties by themselves to correctly fold, to function, to have some enzymatic activity. And also, we'll talk about this later, so no also. Let's then have some idea of current proteomic projects, like well, humans, we are mostly interested in ourselves. So there is a human protein project, which is not quite as successful as human genome project to the date, but there are 3 different groups in this project. One is just a way to get all the protein products for all the chromosomes that are already sequenced and just to catalyze it. We have really nice databases as a result of working in this group, like Protein Atlas. We have all the proteins, and you can actually ask for antibodies for those proteins, and you could get some peptides from these proteins and so on. And there is the second group, which mostly interested in development and disease type of human proteome. They study how proteome reacts on some disease conditions. And there is a third group, which is called Human Resource Pillar Proteome Project, which provides us with needed bioinformatics and other tools to work with proteome. Actually, the problem of being not so successful is problem not of this growth per se, but our perception. Because when first human genome was published, you know that there was a lot of ideas that now we can solve all the problems. We know everything about our humans. There is nothing more. Our classical biology, histology, and everything is outdated. We only need molecular biology and genomics. Then there was less popular idea with transcriptomics, and we haven't have such a big human transcriptome project, right? Then we have human proteome project. We have a little bit less funding, but have produced a lot more data. But each level of organization can only allow us to work with some specific cases. For instance, Human Genome Project. It's fine for some monogenic diseases. You have a gene, there is some mutation or deletion, and voila. transcriptomic project. Not so successful, but still can allow you to see some dysregulation, which also leads to such problems like we already talked. And Proteome project is even more complex. And it wouldn't be possible to get simple answers just today. We're just at the stage of getting all the information needed. But I think it's also nice to visit this site because there is a lot of links to databases that were done with the help of this project. And I think that the main thing that we could use from this project is the infrastructure of databases and the places where you could upload your proteome for others to analyze. Like we have these databases for sequence reading archive, SRI, where you could put your transcriptome genome data. The same is also with proteins. I will be talking about Proteome Exchange website, where you could put all your data, and you could also reanalyze data from other groups as well. So, let's have some view on this human proteome project. We have already been discussing that there are some housekeeping genes and some genes which present only in one copy and only some cells and some tissues. And let's now characterize this proteome from characteristics of amino acids like isoelectric point, molecular weight, hydrophobicity, and so on. What is interesting here is that for hydrophobicity we have almost normal distribution except for this small peak here, which can be explained by proteins which need to be highly hydrophobic to have some portion of this protein inside our lipid membranes. Also, talking about molecular weight distribution, as you can see that most of our proteins are about 40 kilodaltons, not quite big enough, but there are some proteins as big as 310 kilodaltons. But if you are working with proteome, you already know that majority of proteins will be between those numbers. And if you are searching for some biomarkers, apparently it's easier to search this range because there would be a lot more proteins than if you will try to search here. There could be some biomarkers, but the chance of finding them here would be less, right? Also with hydrophobicity here, but with the isoelectric point, it's a little bit different story. As you can see, there are two main peaks, and between them we have this 7.5 pi or pH. Why do you think it is the case? What is a normal pH of most of our bodily fluids?
6.8.
Or up to 7, right? 7.4, for instance. Somewhere in this range. And you can see that this range is actually where we'll have this local minima, right? What do you think about solubility of protein without charge?
There is no solubility.
It's lesser than it can be, right? So if we have most of the protein in this range, most of them will be insoluble. Of course, we need some insoluble protein, like cytoskeleton, for instance, right? If it would be fully soluble, it would be not cytoskeleton. But most of our protein needs to be in solution. So that is why we have those two range here. So already we have some information from our human protein, which can help us with the ranges of those characteristics that we need to work with. Also, those charges here can tell us how likely is it that this protein, for instance, with some basic PI, resides in the nucleus or mitochondria, or in cytoplasm or lysosome. right? So just having a PI of protein is already some predictor of its localization. Of course, there are some notable exceptions to this rule, but as you can see, we can also, by analyzing this human protein, have this nice graph of what proteins are prevalent in which parts of the cell. And what is also interesting, there is a special database, like azymptotic point database. I can just open it also. You'll need it later. But for now, as you can see, at different parts of the cell, different proteins with different PI are prevalent. For instance, if we are talking about nuclear, it's this red one. If we are talking about cytoplasm, as you can see, we have only one big part of it, and so on. So, let's try to see this database. Trust.
What happens in this 7 pH? Is a lot of parts of this cell are not containing a lot of proteins.
You mean here, this
Yeah.
but we've already discussed it. Protein will be not so soluble. If there is a protein with such pH, it is a physiological pH. They would have no net charge. And since there is no charge, its solubility will be lower. And we don't need all the protein insoluble in our cell, right? So only some part of this proteins will be this range. But if we move just a little bit to one or another side, its solubility will be higher, and this way it will stay in our cytoplasm, and they can still function in our cells. So that's why we have such strange distribution of PI here. But let's see this database. There is some mostly used proteins. Let's start with, say, human. This is a distribution of molecular weight per azymptotic point. As you can see, if we'll get those out, we'll have those two peaks. And here you can actually get any protein you want and have this protein all the data on solubility, on its PI, on everything, and also you will get a database of tryptic peptides which can be used to identify those proteins. So quite usable database here. And what we need to discuss at the end of our lecture today is post-translational modifications, because they will alter not only some amino acids residues, but some of those modifications will add or change charge of the proteins, will add or change hydrophobicity, will add or change some other properties. And those post-translational modifications are actually not something unusual. Most of our proteins are modified in one or another way. And as you can see, of course, our proteins are synthesized without any modifications. But during these cycles, already internal modification can happen. Then there is a lot of different modification which can happen by themselves or by special proteins. Like, for instance, we have methionine. Methionine is amino acid which quite easily oxidized. And, for instance, plant use these post-translational modifications, like oxidizing of methionine, like indicator of excessive free radical species of oxygen. So there are some protein or peptides which serves as a sensor of free radicals in plants. So some of modification can occur spontaneously. There is no special proteins which help them to get on other proteins, but spontaneous or just oxidation. And those modifications can be used by itself as a sensors for some kind. Also, to switch on and off some of our enzyme, we mostly need phosphorylation. Because for some proteins to work, phosphorylation should happen before this protein have the conformation that allow it to work. Also, there is glycosylation and some other ways, but all the proteins end up with the same post-translational modification, polyagglutination or polysmolation, which is a tag which says this protein is old enough, we need to recycle it. Then we'll have some degradation by different pathways and get some small peptide of this protein, and then they degrade it to free amino acids. So, as you can see, this post-translational modification in eukaryotic cells is not something that is not usual. It's the usual pathway for protein to be at least degraded. It's a label which says that this protein is old enough, it should be just recycled. Also, some of those modifications can actually predict stage of development. If we just try to see the percentage of proteins which are phosphorylated for plant, then in adult and mature leaves, it will be already decaying, but at later stages there will be less and less and less phosphorylation of proteins. And there's even one theory, I don't believe in it, and actually there's a lot of different articles which says that it's not a case, but there is a theory that aging can be related to lower level of post-translational modifications as well. But there is a lot of theories of aging, and nobody says that any of this theory is the right one. So, actually, not all amino acids gets modified or easily modified, and there are some amino acids which are easily modified by phosphorylation or other. And what we also need to talk about that actually, first of all, post-translational modification is a covalent modification. So there are also different modifications which are not covalent, but it's not post-translational modification. Like two proteins form a complex. Unless there is a covalent bond, it's not post-translational modification. We have two proteins, they interact, but it's no modification. Generally, it is an enzymatic way, so we need some enzyme which makes this post-translational modification, but it can happen by themselves, spontaneous. For instance, methionine get oxidized in any organisms, in prokaryotes, eukaryotes, whatever. It can occur at CO and termini, but it mostly occurs at some radicals of amino acids. And what I want to stress here is that it not only in eukaryotic organisms, it also happens in the prokaryotic organisms. For instance, oxidation, you can't do anything about it, as well as with proteolytic ferments, which just cut some proteins. It's also covalent modification, so it's also post-translational modification. But it is widespread in eukaryotes because eukaryotes have a special organelle for this modification. Can you name it? What organelle is needed and known as a factory of protein modification, protein sorting, protein hiking?
EPS.
Yes, EPS plays a role, but there is one other organelle which is even more involved in it. Golgi apparatus, right. That's why we think that it's widespread in eukaryotes. There is no way to carry out all those post-translational modification in other organisms because they lack this structure. For instance, in bacteria, there is no compartmentalization, and you can't get some part of bacteria with low pH and other part of bacteria with high pH. And for some of these modification, you need extremely low high pH. It wouldn't work in this way, but in our cells, no problem. And plasma reticulum, Golgi apparatus, and we can do it easily. So, let's talk about some of those modifications. And one modification I want to actually to talk a little bit more, and it is glycosylation. It's putting some sugars on some amino acid residues in proteins, because it plays a lot of role in proteins which are receptor proteins, and it also plays a lot of role in our immune system. What do you think? Our immune system can recognize only proteins or also other types of biomolecules?
Other types, of course.
And if other type, which type of biomolecule is a marker of, let's say, a bacterial infestation?
Lipopolysaccharides.
Polysaccharides, right? So that's why actually our cells have a lot of glycosylation events, mostly on proteins which are outside our cells. So you can see here there is a cytosol, there is an exterior part of the cell, and these exterior parts with N-terminus highly modified by those different sugars. Also, primary structure of our proteins can be really the key for them to be placed in some right position. For instance, what if we have, let's say, some protein which have a lot of hydrophobic amino acids, and we have our membrane also here. Let's say we have a membrane, and we have a protein which have hydrophilic, hydrophobic, and hydrophilic part. Will it be enough having this hydrophobic and hydrophilic parts to stay in this membrane in this folding state?
Mostly no.
Mostly no. This can actually, we have two times, three even. If something will be pulling this protein, for instance, is it possible to form some sheet of lipids here and just leave it? Or if you have some micelle which is here, is it possible to actually move this protein from the membrane?
As you can see from the picture.
Actually, there is a problem as it was the last year. I have this presentation in open office format. Unfortunately, there is no open office, and it should be appearing later. So just pretend there is no this picture. It's not here. No, but what we know about lipids in our cells? Do they have any charge in them? What kind of lipids are presented in our bilirubin? Phospholipids, not only phospholipids, but still there are charged groups, right? So mostly we will have negative charge here and here. Also, of course, you can say that there is some membrane potential, which also plays a role, but these short distances will only be interested in those small charges. And if we have some positive charge on our proteins, will it then hamper them in this membrane more readily, right? So it's not only hydrophobicity or hydrophilicity, it's also charge which make those proteins to be correctly positioned in membrane. So let's continue with at least some examples of protein modifications. Let's start with phosphorylation. This phosphate group also brings charge to your protein. And what do you think? How can it influence folding of this protein? Is it a big group? Is it a small group?
It is quite negative group,
Yeah, absolutely.
and therefore it tends to be more active in some cases.
Actually, phosphorylation can switch on or off. It depends on the place of phosphorylation, but it will change the form of this protein. Because why proteins are so widespread and why proteins can catalyze such a lot of different reactions. We have limited radical groups, but what makes it almost indefinitely flexible in catalyzing different reactions?
Different active sites.
Position of those radicals, right? Even if you have only 20 radicals, but some of them charged, some of them hydrophilic, and so on, and you have infinite degrees of freedom, not infinite, but high enough degrees of freedom to just put them in different configuration, you can get the needed environment for reaction to carry over. You don't need pressure, temperature, ultraviolet light, you just position them right. And if it is possible to do the same reaction, because mostly reaction which need catalyzing is something like you have some minima, probably you have some another minima of energy, but you have this hill which need to be somehow get over. And proteins are universal machines in this idea because it can create hydrophobic, hydrophilic, charged, whatever. And it even can fix some water molecule because of charge and position them right in the right way, because water is actually quite reactive chemical if you use it in the right way. And also metals. Most of our proteins use metals to get all the work done, like nickel, even like iron, and so on, so on, so on. And they are quite reactive species. For instance, iron with different level of oxidation. How to say it in Russian? Well, see, yeah, because it's the same. I just forgot it completely. They're absolutely different, and you can modify this by proteins, right? Hemoglobin, for instance, some metalloproteases, and so on, so on, so on, so on. So they can be positioned in different way, and they can use core enzymes or cofactors like some irons and so on to get all this work done. So, just small change in position of this active center can switch it on or off. Or if some substrate is charged and this phosphor is negatively charged, it can prevent or block incoming substrate. So this enzyme wouldn't work because no substrate can get into this active center, right? So those modifications are quite widespread, and if we'll try to see UniProt database, I've just run a small Python script to get this data, and you can see that phosphorylation is quite frequent. Glycosylation, unfortunately, I've divided it into two different types of glycosylation, but if you sum this up, it would be second most frequent. Then it would be acetylation, and so on, so on, so on, so on. You can get this information from this site. They actually have more up-to-date frequency numbers, but still. And let's see some more or less known post-translation modifications, because in textbook ubiquitination is just a label that this protein needs to be degraded, right? But it's only so and only right if we are talking about polyubiquitination. If we have a lot of those ubiquitin, and if they are connected one to another at some certain way, then it's absolutely right, it's a label for protein degradation. But some single event of ubiquitination is actually needed for proteins to form complexes, and it's needed for protein-protein interactions. Also, if we have multiple but different way of polyubiquitination, as you can see, it's also have some role in protein complex assembly and endocytosis. And some other ways of the same modification leads to different outcomes. So for every post-translation modifications, there is a number of different ways how this post-translation modification can work. So there is no one-to-one. There is some ubiquitin, then this protein will be degraded. There is phosphorylation, then this protein will switch on. No, it could switch off, and it could have no effect at all, and whatever. It depends on a lot of factors. So we already talked about phosphorylation, glycosylation. And here comes our Golgi apparatus. It's where all the glycosylation takes place, actually, mostly, and only some initial oligosaccharide can be added at endoplasmic reticle. Ways to add sugars, there is a lot on different ways, through nitrogen, through oxygen, through some other way, and what is interesting is that it is possible to add not only one sugar residue, but a lot of different sugar residue and some other, like phospholipid tail here, ways which have different functions. So we are talking about just glycosylation, but actually it's a lot of different sugars and different sequences of those sugars, and there is such a division of proteomics like glycoproteomics. And there is also one theory of aging which says that it's all because of lacking of those modifications. We'll be talking about theories of aging, I think 3 times more in this course, but this is the second one. So, all those modifications have some correspondence to biological functions. As you can see, there is some mostly usable biological functions and some post-translation modification. And for some modification, for instance, if you have methylation, it's mostly during transcription and chromosome migrants. So if you have this type of post-translation modification, you know what processes this modification can alter. For some modification like phosphorylation, of course it's not as obvious, there is a lot of different processes, but there is still some correspondence between some modifications and what mostly it will change. And to end today's lecture, I have time already, I also want to stress that not only proteins matters in biomedicine, but also post-translation modifications. For instance, of course, Alzheimer's disease starts not with tau protein, but tau proteins actually the one that gets the most damage to our nervous system. And this protein accumulates only if there is a lot of phosphorylation done on it. So if there is no such post-translation modification, there would be no tau plaques. Also, there is some cardiovascular diseases which are characterized by phosphorylation of different proteins, and if there is no such phosphorylation, there would be no disease. If it starts with phosphorylation, it's probably the problem. Some different modification manifests different problems, and also we need to talk just for 1 second about glycosylation. You know that our immune system don't like different xenobiotics, and if they have big molecular mass, we can get allergy or even immune reaction to them. And we can mask some of those epitopes by adding glycosylation to them. So we can use some proteins which are not present in our body and mask them with glycosylation. Unfortunately, the same is true for viruses, which also tries to use our own machinery to mask some antigens. Our coronavirus is one of them. So it's actually worth studying. I think it's all for today. If you have a question, just ask me. If not, we'll continue tomorrow.
Is there a way to evaluate the level of phosphorylation for each of the protein?
Yes, there are some chromatography phases which can enrich your phosphorylation proteins. So all the proteins with phosphorylation will stay on this column, chromatography column, and you can elute them later. So you'll only get fraction with this modification. With, for instance, titanium oxide columns, it is possible to do. There are some other chemistries. So it's possible to actually research only some post-translation modification from the whole proteome.
When should I do this? When should I do this?
When?
20 years later, I'm working out.
You mean this one? You know, brain biopsy is not a way to live, so it's better not to do so. There are a lot of other biomarkers which can help to try, but actually, since I came from neurobiology and I work with some neurodegenerative diseases, I want to say that it's not only the progression of disease per se, but plasticity of brain. And there are some correlation between some habits and an onset of Alzheimer, even in some cases where it is hereditary, for instance. So having to know about 1 or 2 languages aside from a mother tongue, having playing musical instrument, not relying on maps or phone books, and so on, have some correlation. It won't 100% prevent from this disease, but it can get onset of these diseases to longer age, so it wouldn't happen. Because what I have in my laboratory, it is possible to see the brain with, which definitely have this tau protein tunnels in here, dilated vesicles, thin cortex, and there is a medical record states man 95 died from cardiac attack, have no cognitive deficits. And there's also a problem when you have some samples, human tissue, when there is no visible damage, absolutely. Of course, when you do some molecular biology, there are some small negligible results, but the card states that patient died from some trauma that was that he get because of his cognitive deficits, and that it says that some I don't know, some person 40 serious cognitive deficit, diagnosed with Alzheimer's diseases and so on and so on, but so it's not one-to-one patient. I think there would be some problems with our coronavirus because it actually have some tendency of amyloidogenesis, not prions per se, but you know amyloids and prions are close enough. there are ways of so-called physiological amyloids. For instance, some of your cells produces peptide hormones, and those hormones need to be stored in the cell in really high quantities. What would happen to the cell if it will have all those peptides in cytoplasm in soluble form?
They start forming other peptides.
Yeah, but what if you just change this peptide a little bit, that it can form those aggregates, but those aggregates can be easily disassembled by pH or something when the cell actually needs this hormone? Will they be troublesome if they are insoluble?
Not so much.
Not so much, right? So amyloids is not only pathological form. Other problem, biofilms that generated by bacteria. Most of them have those amyloid proteins. It's mostly proteins with beta sheets, and those beta sheets just stacked one on another. And for bacteria, it's really a good idea to have this biofilm. It just releases all the protein on the outside, and it absolutely, without anything, just combined to this amyloid form, and it can help bacteria to survive, because nothing can eat through this biofilm, which is a problem for us people. Also, just 3 years ago, there was publication on plants, actually just our pea plant, our garden pea, which we use, which we eat in canned form, in other ways, have some protein which is stored in this pea in amyloid form, which prevents fungi to grow. For fungi, amyloid is toxic, and fungi can grow on these products, so it's also possible. So amyloids and prion a little bit different, because prions are amyloids, but not all amyloids are prions. Prions is amyloid which have tendency to make other proteins fold in the same way. So they are, let's say, chaperones that just copy themselves. So other proteins have the same folding state. Amyloid can be those chaperones, but they do not have this this determinable effect. It's mostly physiological. Any other questions? You can ask questions tomorrow. We'll have shorter lecture, and I'm afraid that we'll finish earlier. So if you have any questions, we are welcome tomorrow.
Thanks a lot.